Welcome guys, we are learning about the 1H NMR spectroscopy. In previous lecture, we learned about the spin state of the molecule. So, in spin-spin coupling, there is a spin relaxation time. So, it is a longitudinal relaxation and the transverse relaxation. In most spectroscopic techniques, how the energy absorbed by the sample is released is not a primary concern. In NMR, where the energy goes and particularly how fast it gets there are the prime of the importance. The NMR process is an absorption process. Nuclei in the excited state must also be able to relax and return to ground state. The NMR process is an absorption process. Nuclei in excited state must also able to relax and return to ground state. The time scale for this relaxation is crucial to the NMR experiment. For example, relaxation of electron to ground state in UV visible spectroscopy is very fast process on the order of the picosecond. In NMR, the excitation state of the nucleus can persist for a minute because the transition energy between spin levels is also small. Attaining the equilibrium occurs on the much longer time scale. The time scale for relaxation will dictate how the NMR experiment is executed and consequently how successful experiment is the. There are two processes that achieve this relaxation in NMR experiment. Longitudinal, that is spin lattice relaxation, and the transverse, that is spin relaxation. So here we are going to discuss about the longitudinal relaxation. In longitudinal relaxation, energy is transferred to the molecular framework, the lattice, and the lost as a vibrational or a transition energy. The half-life for this process is called the spin lattice relaxation, that is T1. Dissipating the energy of NMR's transition into the sample should not be the problem. However, T1 values are often long. The problem arises not in where to the send the excess energy, but the pathway along which the energy is released to lattice. Contributing factors to this type of relaxation are temperature, solution viscosity, structure, and molecular size. So now we are discussing about the transfer, that is spin relaxation. So in transverse relaxation, energy is transferred to the neighboring nucleus. The half-life for this process is called the spin relaxation time, that is T2. The process exchange the spin of nucleus A with the spin nucleus B. There is no net change in spin process. In homogeneity of the magnetic field or the presence of paramagnetic material can be a large contributor to the value observed for the transverse relaxation. The peak width in NMR spectrum are inversely proportional to the lifetime and depend on both T1 and T2. Most organic solution T1 and T2 are long enough to result in spectra with sharp line. However, the magnetic field homogeneity is poor or the paramagnetic material such as iron is present in NMR signal can be broadened to the extent that the signal is destroyed or the unusable.